There we go. Okay, that wasn't too bad. So if you just picked up this recording, there's a list of characteristics of specific adaptive immunity, and you'll want to get that from a person that was in class. Get those notes. If a person develops specific immune responses, that happens in a particular pattern. And this pattern changes based on whether it's your first or your second or 12th exposure to that pathogen. And we call this primary and secondary responses. There should be a word right here, primary, in this space. So like this. So I messed up making the slides. So it should say result in primary and secondary responses. And then your third blank is the word antibodies. This graph, we have a version of this graph at the end of this section. And every test you have a graph that you have to draw the graph. And it's the big concept stuff in immunology. This is the number one. If you only remember one long-term, make it this. So we're going to spend some time looking at this graph and relating different immune functions to it. So this will be the one that you draw on exam 4. Draw the graph that characterizes primary and secondary immune response. And the y-axis on this is how much antibody. How much antibody are you making? And then the x-axis is time. So we're looking at how much antibody do you produce over time. And this is not just all antibodies that you can make in total. This is antibody against a specific pathogen. And this will make more sense in a minute when we talk about what antibodies are, but you've probably heard of antibodies. This is a measurement of antibodies, but it's antibodies against a specific pathogen. And we normally measure these in plasma or in serum. And it can also be known as the antibody titer. T-I-T-E-R, antibody titer. Around here, people say titer. That's what that means. It's a concentration of antibodies. And this starts when you are very first exposed. So when you have an initial exposure to that very specific pathogen. And we'll say as an example, COVID-19 Omicron. So not just COVID virus generally, and not just virus generally, but a very specific virus like COVID-19 Omicron. And then from that point that you're exposed, you start to, after a while, you start to create antibodies, and those levels kind of rise. And they rise kind of slowly, and then those antibodies will persist for a while, and then they'll kind of decline again. And this first response is called primary response. Okay, and on this graph, you've got this graph. Make a note that this time frame right here would be a totally variable amount of time. This graph is making it seem like immediately the person got exposed again to COVID-19 Omicron, which is realistic actually. But this wouldn't have to be the next day. This could be 2 years later or 17 years later or something. You get exposed again. And we call that the secondary exposure. A secondary exposure has to be to the same specific pathogen. Put the same in quotes. Needs to be the same thing, not a different thing, not a mutated thing, the same one that you saw before. And if you have developed specific acquired immunity, the next time that you are exposed to that same thing again, you suddenly get a much quicker and larger production of antibody. And we call that the secondary immune response. So this is a faster and stronger antibody production. Faster and stronger. We're going to come back to this every so often in the slide set. So add some more things after we talk about, okay, what are the antibodies and how do they act and how does all of this work? I really like OpenStack's pictures about this particular topic. They're very good. So we've mentioned a couple times in different ways that this requires exposure to a specific pathogen. And that pathogen is something that is got characteristics that aren't us. So it's a foreign object and it's not part of ourself. So there's this concept in the immune system of you've got your self shapes and then you've got your foreign shapes that are not part of yourself. And we call those shapes antigens. If you've not been in AP2 yet, this antigen might be a new word. So what an antigen is just a small 3D shape. So maybe write that off to the side of antigen. It's a small 3D shape that's capable of causing immune response. Or let's say specific adaptive immune response. It's a little shape. And on that antigen, there can be even smaller little individual shapes that are called epitopes. And so this picture is representing the whole bigger 3D shape that could cause an immune response as the antigen. But there's some little smaller pieces of it with their own individual shapes called epitopes. So if I wanted to recognize Hunter, like Hunter could be an antigen, but there's certain parts of him that I would recognize. Oh, I know that is Hunter because he has that beseech shirt that he wears a lot, or those certain shoes, or like your haircut that you have right now, which is different than last year. Like that would be an epitope. Like my hair is a big time epitope for people to recognize me. I went to band competition for NOAA this weekend and someone recognized me from 50 yards away from high school. I haven't seen them in a long time. And then I heard their voice behind. They were walking with another friend. They're like, yep, that's Laura. I turned around. It was my friend. My hair is my recognizable epitope. So any foreign shape antigen is going to have multiple little pieces on it called epitopes. And the epitopes are actually what our immune system recognizes. So go back to here. When you had specific pathogen, and you could add in the word epitope. So any place where we've had the word specific pathogens, you could add in there, it's the epitope, the little shape that's being recognized. And for COVID infections, the major epitopes that we recognize are spikes, the spike proteins. And the reason that COVID keeps kind of making people sick is that the spikes change shape and our body can't tell that's the same thing. Because there's a little shape change. It's like if I want to hide from people, I can put on a hat and no one will recognize me unless they know me real well. If you ever see me in Virginia Ave Food City with a hat on, I'm probably trying to be incognito, but I'll still talk to you if you recognize me. So our body recognizes these shapes to know what's us and what's not us. And our shape recognizers in the body are called antibodies. Antibody. So see on this cartoon here, they've got these kind of Y-shaped looking things, antibodies, and the antibodies are binding onto epitopes, and they've got it color matched. This is a really good picture. So if this is our foreign object, and it's got 3 types of epitopes, each epitope would be recognized or bound to by a specific antibody that just recognizes that. So this one recognizes orange circle. This one recognizes red cone. This one recognizes green box. And antibodies recognize epitopes by binding onto them. And just by binding, that's helpful in a lot of ways. If we can bind antibodies onto the foreign shapes, we can eliminate them and their antigen and their microbe in a bunch of helpful ways. This is kind of what the structure of an antibody is, and we're going to write a bunch of things about this. A former student got me this antibody doll. Kind of obsessed with it. I really love it. And it's got all the different parts of the antibody in a cute and non-threatening way, which is better than these kind of cartoons. It's got all the parts. So I think about antibodies as binders of shapes, and this is the shape here. This would be an epitope. It's kind of cross and dangerous. And the only thing I would change about this antibody doll, it's not totally perfect, is that there should be 2 epitopes. There should be one for over here also, because whatever antibody binds with this half, it would bind the same exact same thing on the other half. But I don't want to buy a whole separate one of these just to get another epitope. So we could call this the epitope, or we could call this the antigen. But the antibody is symmetrical, and it has parts that bind the epitope, and then it's got parts that do something else. It does not really have eyes, just to make it cute. So antibodies are proteins. You should write that down probably in this space. They are proteins, and we build these proteins in a very interesting way. But the other name that they can have is immunoglobulin, or IG. So in this space next to the word immunoglobulin, write capital I and lowercase g. That's the abbreviation. People thought of the concept of antibody before we could do protein chemistry and even discover proteins. So the word antibody is a real old word. And then later it was discovered that in people's blood, they had this set of proteins that were blobs, that's the globulin part. And they identified a bunch of these globulins that seemed helpful for immunity, and they called them immunoglobulin, which is abbreviated IG because that's just a mouthful. So immunoglobulins have this particular conserved shape with different regions, and I'm gonna have to erase these from the graph, and then we'll go back to the graph, and that'll be okay. I'm gonna draw some stuff on this picture. Okay, so these are proteins, and this is kind of a generic one. And we make these antibody proteins out of 4 pieces. So they have 4 pieces. 2 are called heavy chain, and that's in the purple here. So this is 1 piece of heavy chain. This is the second piece of heavy chain. And then the other 2 pieces are called light chain. And they are separate pieces of protein. And you've got 2 of them. This whole thing is symmetrical. So we build these out of matching components, let's say. So you have 2 of the exact same type of heavy chain, 2 of the exact same type of light chain, and they are joined together by disulfide bridges. So this antibody is kind of very solidly put together, but I know why they made it this way, because kids would rip it apart if you actually played with it, if it was just held together by these disulfide bridges. That's not so important for this class. What is more important for this class is what the ends do. So you kind of have 2 kinds of ends. You have an antigen grabbing end or antigen binding sites. Those would bind the epitopes, the foreign shapes. And then this part down here, that's kind of like the, I don't know, the nose or legs, I don't know. This part can either bind to cells or cell receptors at certain times. And those ends of the antibody are called FC. The FC end, it stands for fragment conserved. That's not important for this class. Versus the fragment antibody binding. There's going to be some places where we see FC coming up in some future slides. All that means is this part of the antibody down here where the tag is. That's the FC. Last thing before we move off of this slide is that a real antibody kind of has some wiggle to it. This is the true protein shape, and you can kind of see that there's a real narrow section there. So a real antibody can kind of be like doing like this all the time. And it's got some flexibility to it when it binds these foreign shapes. So what we're going to look at next are some terms that have to do with, okay, so antibodies can bind foreign shapes, and this is helpful. How is that even helpful? This is a list of helpful antibody actions. And the first one is just called neutralization. And we can see on here like here's a virus and it's got spikes sticking out. And if antibodies can match to those particular spike shapes or those particular epitopes, they could bind enough spikes, kind of cover them. And they might be able to prevent those spikes from attaching to the cells. That's neutralization. In this case, the antibodies are binding onto a section of a toxin and covering up the harmful part of the toxin. That's neutralization. And in this case, this makes me laugh. The antibodies are binding enough onto the flagellum to keep it from waving and swimming effectively. This one's kind of silly to me. This one's very important. So when we first started, scientists first started testing COVID vaccines, they were very interested to know, okay, if we give people a COVID vaccine, are they going to produce neutralizing antibodies in levels enough to prevent the virus from infecting more cells? That takes a lot. Like if the virus has 8 spikes, like 8,000. But see how this one is covering up some, but not all? It could still bind with some of these. So this takes a lot of antibody to do neutralization, but that's a really helpful method. There's other ways that antibodies can help block infections. And the first is called agglutination. Agglutination is like clumping, and agglutination depends on an antibody having multiple places where it could grab with one side, it could grab something else with the other side. Clumping different pathogens together and preventing them from moving. So this clumping action can prevent pathogen movement. And notice on this picture that these antibodies look different, like a snowflake or something. This is an arrangement of antibodies in a pentamer. So it would be 5 of this dude kind of linked together at their feet, and together they could bind 10 of the same thing. We'll see what that type of antibody is called in a minute, but the more grabbers, the more antigen binding sites you have, the better you are at agglutination. Another way that antibody binding helps is that if antibodies bind on the surface of a bigger pathogen and there's a bunch of them on there, together, their conserved region, the FC region, there are receptors for those on macrophages. And the favorite thing, or the most tasty looking thing for a macrophage to attack and eat, is a pathogen with antibody all over it. That's called opsonization. So macrophages already like to detect these pathogens by their PAMP shapes and engulf them. But if they've got antibodies all over them, that will happen a lot faster. So antibodies make pathogens more recognizable for phagocytes to eat them more quickly. That's a helpful way. Yes, these would be methods 4 and 5. Antibodies can help activate complement. So complement, that was an innate thing, right? Complement actually works in both innate and adaptive branches. So antibodies binding onto the surface of a pathogen will encourage complement cascade activation to more quickly put these membrane attack complexes into a cell and lyse that cell. So complement will work without antibodies, but if antibodies are present, this will work faster and better. And finally, antibodies on the surface of a pathogen can also encourage natural killer cells to more quickly attack and destroy this cell. So that's a role called antibody-dependent cell-mediated cytotoxicity, ADCC. ADCC responses are basically, we have antibodies are going to encourage NK, natural killer cells. And one we haven't talked about yet, but TC or T cytotoxic cells to work better. It's kind of like if you want a team to work better, bring them food. Like, they're going to do the same work, but they'll do it faster and better if you've got a lunch or something to urge them on. So those are the five helpful things that antibodies do. And antibodies occur in 5 varieties as well. And this same chart is in every micro book, like no matter the level, and it's got basically all the same information. This information for you all is not equally important, but some of it's more important than others. So I want to use this chart to circle or highlight what is most important in this. So what we have across the top are varieties of antibodies, and you notice that 3 of them look basically like this. You got the 2 antigen binding sites and the 1 FC conserved region. But there are 2 that have a different configuration. So all of these can bind antigen. They are all built basically the same way, but some of them are have more components than others. And they have particular names. So they're called IG something. So let's circle those. There is IgG, immunoglobulin G. There's one called IgM. That's the one in the pentamer, like a snowflake. There's one called IgA. It's 2 together, feet to feet, sort of. There's an IgD and there's an IgE. The next thing that I want you to highlight or probably underline is that for IgM and IgA, those are not just a single antibody, they have multiple together. So IgM is pentamer. It's 5 antibodies grouped together, cooperating. It's a pentamer. And because of that, it has 10 antigen binding sites, because every individual has 2. And then likewise, this IgA that's 2 antibodies linked together, it's a dimer. two of it together. And because it has two antibodies linked together, it's got 4 antigen binding sites. All the others are just on their own and they have 2 antigen binding sites. Okay, so next I'm going to mention some particular things about these. So the most important probably of all of these antibodies is IgG. And you'll notice that is the one that is the most present in plasma. So this, how much is in the serum? That's talking about in the plasma. And most of your floating around in your plasma antibodies are in the form of IgG. IgG, I feel like for what you all are going to learn about in nursing school, that you need to know what that one does the most. And it does most of the antibody things. And it's especially important that that's the one that crosses placenta. This one, the mom can pass it to the fetus before it's even born, because this has like special clearance to go through the placenta and cross into the fetal circulation. None of the others can do that. And that's all the things I want you to know about IgG. The next one that's pretty important is IgA is what's called secretory. Secretory. And I don't know where I want to write this. Maybe just off to the side. IgA is known as secretory. Not secretary, but secretory antibody. And we put that into body secretions. So it would be found in saliva, in mucus, in breast milk. What else? All the different mucus everywhere in your body. I guess those 3 things will cover it, but any secretion that you make, if it can have antibody in it, it's only IgA. That one goes into secretions. And then for IgE, I can't say it, IgE, letter E. Is involved in our immune system attacking worms. So not worms attacking you. I guess they would have to start attacking you first, but our immune system attacking worms, and also in allergy responses. Those other ones you can get by pretty fine without knowing their specific roles. If you later want to do PA school or something like that, keep this slide. It will be useful to you in the future if you need to go back and know extra things about antibody function. And that reminds me, all of you should keep these slides, because I try to make them like, here's your core study stuff for nursing school or for PA school or PT school. Those of you going to PT, you don't need immune stuff, but like for A&P2, hope you kept those. But those are your antibody classes. This, just in case you need this for other future upper level classes sometime. These are the Greek letters that match this name. So this is gamma for like IgG. And this is mu, like for M, IgM, alpha, delta, epsilon. So journal articles use those letters all the time. And if you don't realize, oh, that's what that means, it seems like a whole new thing, but it's not. they match the whatever our alphabet is called. So antibodies are helpful, and we make 5 categories, and they can do 5 helpful things. And they are doing most of the work in specific adaptive immunity that we call humoral. And this is not like funny hubris, but humoral immunity. And humoral immunity comes from, we've got soluble antibodies floating through fluids. So that's the immune system that's helpful in blood and in tears and mucus and saliva and breast milk and amniotic fluid, all the fluids of your body. That's humoral immunity. We have another branch of specific adaptive immunity called cellular immunity. And so cellular immunity comes from immune cells directly attacking other cells. So it's kind of like, is the immunity most helped by antibodies, or is the immunity most helped by cells? And that's called cellular immunity. To go back to COVID vaccines example. COVID-19 vaccines. When they were testing how well do COVID-19 vaccines work, they could measure different things. So we can measure the antibody levels in the blood and measure pretty easily, like how well is humoral immunity working. And that's easy and quick to measure. And that's also the general public hears that and like, do I have that? Do I have the antibodies? People come up and ask me like, you think I have the antibodies if I got the vaccine? And like, yeah, you probably have some level of antibodies. But also COVID vaccines, if they work well, they also ought to produce memory cells and something called cytotoxic T cells that can attack viral infected cells. This one's harder to measure. This one's very expensive to measure. And because we needed vaccines really quickly, they're still like trying to figure out how well did the memory cell and T cell responses, how well did the cellular immunity work from these vaccines? They probably worked pretty well because the antibody levels were not that great, but the vaccines were very good at protecting people from getting deathly sick. So probably the cellular part of that response. So now we'll look back and we're gonna look at what cells make antibodies and what cells do the cellular immune response. And it's the lymphoid lineage. So we've got this is kind of the ancestry of all kinds of blood cells. And they come from the bone marrow. And they're kind of in 2 big families called myeloid and lymphoid. So the ones that are most involved in acquired specific adaptive or acquired immunity are the lymphoid cells. So we're going to zoom in on these. Oh, notice something. There should be a T right here, letter T. This is what happens when you make slides at like 10 o'clock. Yep, looks good to me. And the next day, there's this little thing missing. T, letter T. So we make all different types of lymphocytes in the bone marrow. We make these in the bone marrow. They're specific white blood cells. And all of those lymphocytes, depending on where they go in the body and what genes they decide to use, they will either become what we call T lymphocytes or B lymphocytes. These, a lot of books and people would call them T cell and B cell. But they are technically the blood cell type called lymphocyte. And whether you are a T lymphocyte or a B lymphocyte, you are kind of develop over time the ability to bind to one specific antigen shape. And you will have receptors coming out of your surface. Let me make them look a little bit different than antibodies. They're not antibodies on the T cell. So this is a receptor on the surface of the T cell that can bind onto a specific antigen. And this is called T cell receptor. It would bind onto a very specific antigen shape. The B cell family also has specific receptors, but their receptors actually look like antibodies, and they basically are antibodies. So this is called B cell receptor. And depending on the type of B cell, this can be an IgM or an IgD. So IgD is an antibody that basically never floats around. It just stays in the surface of the B cell and helps it bind to its antigen. So being a T cell or a B cell is kind of a depressing life, because what happens is they form, they have to travel to a specific place to become mature. So let's add that on here. Where do they mature? On red for this. Where do they mature? Aaron, where does T cell mature? They go to lymph nodes for round 2. Where's the first place they go? They're a T cell. Thymus. Yeah, which is one of those organs people never think about much. So if you're a T cell, you mature in the thymus after you leave the bone marrow. And what happens during that process is you get your special receptor set. If you're a B cell, you mature in the bone marrow. And so when they're undergoing that process, what I mean by mature is that they get their final receptor set, I'm gonna call it. When they're first formed in the bone marrow, they don't have those receptors yet. They don't have their binding specificity. So we can make millions of kinds of these, and we make random binding specificity so that any foreign shape that comes to us from anywhere, you could bind it. So imagine that you are a T cell and you finish your training and you have a specific T cell receptor shape that lets you bind smallpox antigen. And you spend the rest of your life drifting around the body, holding your receptor out like this and waiting to touch smallpox. And if you live in this century, you will never find it, and you float around until you die, until the person dies, and you never touch your antigen enemy. And that's a good thing. Like, don't need to fight that off today. But that was your binding. Yeah. Imagine now that you're a B cell and your specific antigen specificity that you were developed with was for COVID-19 alpha variant spike. And you float around with your receptors like this, waiting for that to bump into you. I got it. And then I get activated. So this row, moving on to the next thing. Just because you exist and you have that specificity doesn't mean that you are activated or have activation. We need one more relevant fact before we talk about activation. So we make tons of lymphocytes, B and T cells, so many. But we make just a few B cells and T cells. With a binding ability for any specific epitope. So when COVID viruses first swept through humans, there are humans that had B cells and T cells to detect COVID antigen shapes, but there might be like 4 to 9 in their whole body. And those 4 to 9 are enough to detect that the shape is here, but they are not on their own enough to do anything about it. They can just detect that it's there. So what you need to get actual strong immunity is to get these cells activated. And they are activated by binding their special antigen. And that's where we'll stop for today. They're binding to their specific antigen, and then they get activated. And then immunity can start. That's where we will pick back up on Wednesday. As you're studying for lab exam, what was your all's last slide today? Is it this? Yes. This one right here? Good. I started building out the next set. If you have questions about lab exam the next couple days, email me or come by and see me. I mean, it's tomorrow. If you have any questions about the lab exam in the next set of hours, let me know. And I'll see you for lab tomorrow.